A stone slab opens in northeastern China. Around the skeleton of a small dinosaur, there is a faint halo of fibers. They are not plant remains. They are feathers. If feathers existed before birds, what else did evolution build before its modern purpose? 10 discoveries would force scientists to rethink the history of animal life. Number 10. Kimberella. More than 550 million years ago, the seafloor looked almost empty. There were no fish swimming above it. There were no crabs crossing the mud. There were no shells scattered across the bottom. Instead, broad microbial mats covered the sediment like a dark living carpet. Across one of those surfaces, something moved. Its body was soft. It had no bones and no mineralized shell. When it died, almost nothing should have remained. Yet, in rocks near Russia's White Sea, paleontologists found flattened impressions of an oval organism now known as Kimberella. The earliest reconstructions made it look like a jellyfish. That interpretation was understandable. A soft body pressed into sediment loses its original shape. The upper and lower surfaces collapse together. Features that were once three-dimensional become faint lines on a flat slab. With only a few fossils, researchers could not easily separate real anatomy from damage caused by burial. Then more specimens appeared. Not one or two, but dozens. Some were broad and oval. Others were narrow. Different fossils preserved the body in different positions, giving scientists more angles from which to reconstruct it. Kimberella no longer looked like a jellyfish. It had a recognizable front and back. Its left side broadly mirrored its right. That bilateral symmetry was important. Most complex animals alive today share the same basic arrangement. A front end encounters the environment first. Sensory organs can become concentrated there. Muscles can be organized for movement in one main direction. Kimberella appeared to possess that type of body plan before the Cambrian period had even begun. Its shape was only part of the evidence. Near some fossils, researchers found fan-shaped marks cut across the microbial surface. The marks had been known for years, but the animal responsible for them was uncertain. Their size and position suggested a connection with Kimberella. The animal may have moved across the seafloor while scraping or raking food from the microbial mat. If so, the rock had preserved more than a body. It had preserved behavior. An animal had moved across the mud, fed, and left a record of that activity more than 500 million years ago. Kimberella was not necessarily a modern mollusk in primitive form. Its exact position on the evolutionary tree remains debated. Some features are consistent with an early mollusk relative. But the animal also differs from anything living today. The stronger conclusion is broader. Complex, mobile animals with a clear left and right side may already have existed before the sudden abundance of shells and skeletons seen during the Cambrian period. The Cambrian explosion may not have marked the true beginning of complex animal life. It may have marked the time when older lineages became more diverse, more widespread, and easier to preserve. Kimberella pushed the visible story of complex animals farther into the past. But its identity still depended mainly on shape and traces in sediment. Another Ediacaran organism was even more difficult to classify. It was flat-ribbed and unlike any animal alive today. To identify it, scientists would need evidence smaller than any bone or footprint. They would have to search for molecules trapped inside the fossil itself. Number 9. Dickinsonia. Dickinsonia looked like a ribbed oval pressed into stone. Some specimens were only a few centimeters long. Others reached more than a meter. Its body was divided into repeated units extending away from a central line. Depending on the specimen, it could resemble a quilted leaf, a flattened worm, or a pattern folded into fabric. None of those comparisons explained what it truly was. Dickinsonia had no obvious head. It had no limbs. It had no skeleton. It had no preserved mouth that every researcher accepted. It had no internal organs that could be identified with confidence. For decades, scientists placed it in almost every major biological category imaginable. Some considered it an early animal. Others proposed that it was a giant, single-celled organism. It was compared with fungi, lichens, and microbial colonies. Another possibility was that Dickinsonia belonged to an extinct branch of complex life, with no surviving relatives. Its shape could not settle the argument. A flat impression might show the outer form of a body, but it could not always reveal the biology that once operated inside it. Then, unusually preserved specimens were found in northwestern Russia. Instead of surviving only as impressions in sandstone, these fossils retained a thin film of dark organic material. The material had been altered by burial and geological time, but parts of its molecular composition remained. Researchers removed tiny samples from the fossil and from the surrounding microbial surface. They were not looking for DNA. DNA breaks down too quickly to survive for hundreds of millions of years. They were searching for more durable organic compounds, including the remains of sterols. Sterols are molecules used in cell membranes. Different groups of organisms tend to produce different mixtures. Animals are strongly associated with cholesterol and related compounds. Plants, algae, and fungi usually leave different chemical patterns. When the material from Dickinsonia was analyzed, it contained a high proportion of cholesteroid molecules. The surrounding microbial mat did not show the same signal. That difference mattered. The molecules were not simply coming from organic material spread throughout the rock. They were concentrated where the body of Dickinsonia had been. The fossil carried a chemical signature consistent with an animal. After decades of debate based mostly on shape, the identification had gained an independent form of support. Dickinsonia was not merely an impression that happened to resemble an animal. Its preserved chemistry indicated that it had been one. The result was powerful, but it did not solve every mystery. The molecules did not reveal whether Dickinsonia had muscles arranged like those of living animals. They did not identify its closest relatives. They did not explain how it fed. Some fossils suggest that individuals moved across microbial mats and left disturbed patches behind. That may point to absorption or external digestion, but the exact process remains uncertain. The chemical evidence answered one question while sharpening several others. That is often how paleontology advances. A discovery rarely produces a complete picture. It removes some possibilities, it strengthens others, and it changes which questions matter most. Kimberella and Dickinsonia showed that recognizable animals existed before the Cambrian period. One was understood through anatomy and traces of movement. The other was identified through preserved molecules. But recognizing early animals was only the beginning. As animal bodies diversified, evolution began producing shells, limbs, jaws, ears, wings, and feathers. Some of those structures are so integrated that they appear impossible to imagine in an incomplete form. The shell of a modern turtle is one of them. Its ribs, spine, shoulder girdle, and skin are reorganized into a single protective system. For years, even the oldest known turtles already had nearly complete shells. Then, a fossil from China revealed an animal whose armor was only partly assembled. Number 8. Odontocules. A turtle cannot leave its shell behind. The shell is not a separate covering placed over an ordinary skeleton. It is part of the skeleton. The upper shell, or carapace, is connected to widened ribs and vertebrae. The lower shell, or plastron, protects the belly. Even the position of the shoulders is unusual. In most land vertebrates, the shoulder girdle sits outside the rib cage. In turtles, it lies inside the structure formed by the ribs and shell. This arrangement made the origin of turtles difficult to reconstruct. For many years, the oldest known turtle fossils already possessed complete armor. They were ancient, but they did not show how the shell had formed. One idea proposed that the shell began as small plates of bone in the skin. Those plates may have expanded, joined together, and eventually fused with the internal skeleton. Another possibility was that the ribs themselves broadened first, gradually changing the architecture of the torso. The fossil record offered little between an ordinary reptile body and the highly modified turtle condition. Then, paleontologists studying Triassic rocks in southwestern China described an animal about 220 million years old. It was named Odontoceles semitestacea. The name roughly means toothed turtle with a half shell. The animal had a broad body and several characteristics linking it with early turtles. Its armor, however, was incomplete. From below, Odontoceles looked surprisingly turtle-like. It had a well-developed plastron covering the belly. From above, the picture changed. Its ribs were widened. Bones along the spine showed changes related to shell formation, but a complete carapace did not cover the back. The upper shell had not yet reached the condition seen in later turtles. Odontoceles also had teeth. Modern turtles have toothless beaks, but this early relative retained a more ancestral jaw. The combination was revealing. A recognizable lower shell existed alongside a back that was still undergoing transformation. The fossil suggested that the plastron formed before the carapace was complete. It also supported the idea that the upper shell developed through changes to the ribs and vertebrae, rather than simply from armored plates spreading across the skin. At first, Odontoceles appeared to preserve the missing beginning of the turtle shell. Then another question emerged. Had the lower shell truly evolved first? Or had the upper shell become reduced because this animal lived in water? If Odontoceles descended from ancestors with more complete shells, its anatomy might represent specialization rather than an intermediate stage. The argument could not be settled by one fossil. Then, an older animal was discovered in Germany. Papocheles did not have a true turtle shell. Its ribs were broadened and shaped differently from those of ordinary reptiles. Along the belly were thickened gastralia, sometimes called abdominal ribs. These bones had not fused into a plastron, but they suggested a stage before the complete lower shell of Odontoceles. An even earlier animal, Unatosaurus, possessed wide T-shaped ribs and changes to the muscles of the trunk. Together, these fossils created a sequence of anatomical conditions. First, the ribs broadened. Then, bones along the belly became enlarged and organized. Later, a plastron formed while the carapace remained incomplete. Finally, the upper and lower shell became integrated into the turtle body. This was not a straight line, in which one named species transformed directly into the next. Unatosaurus, Papocheles, and Odontoceles were separate branches, but each preserved a different combination of traits, allowing researchers to reconstruct how the shell could have been assembled. The process affected far more than armor. As the ribs expanded, they changed breathing. They altered the attachment of muscles. They transformed the position of the shoulders. A structure that appears unified in modern turtles was built through changes spread across much of the body. Odontoceles showed evolution constructing anatomy in stages. The next discovery revealed the opposite process. Structures that were once essential can become smaller, lose their original role, and eventually disappear. The animal already had the long body and flexible spine of a snake. Yet near its tail were two recognizable legs. Number 7. Najash. Modern snakes are defined partly by what they lack. They have no visible arms or legs. Their bodies are extended into long chains of vertebrae. Movement comes from waves of muscle passing along the ribs and spine. The skull is modified so the jaws can open around prey wider than the head. Snakes did not begin that way. Their ancestors belonged to a larger group of four-limbed reptiles. Some living snakes still preserve traces of that history. Pythons and boas possess small internal remnants of a pelvis. Certain species also have tiny external spurs near the base of the tail. These are associated with reduced hind limbs. The remnants show that legs once existed, but they reveal little about the stages through which they disappeared. Early fossil snakes complicated the question. Several species found in marine sediments retained hind limbs. That led some researchers to propose that snakes originated from aquatic reptiles. Under that interpretation, the body became elongated, and the limbs were reduced as the animals adapted to swimming or moving through marine environments. The evidence was incomplete. Then, paleontologists working in Patagonia found a snake from a very different setting. It lived around 90 million years ago in a terrestrial environment. The animal was named Najash rheonegrina. Najash had the elongated body of a snake. Its skull and vertebrae placed it within the early history of the group. Near the rear of the body, however, was a pelvis connected to the spine. Attached to it were two external hind limbs. These were not tiny fragments hidden inside the body. They included recognizable leg bones projecting outside it. The pelvis was also linked to a sacral region of the vertebral column, a feature inherited from four-limbed ancestors. In modern snakes, that connection is gone. Najash preserved a stage in which the hind limbs were reduced but still integrated into the skeleton. Its environment was equally important. The surrounding rocks had formed on land. This was not a specialized marine snake. Its anatomy supported the possibility that major stages of snake evolution occurred in terrestrial or burrowing ancestors. Additional Najash skulls later added more detail. The skull combined snake-like flexibility with several features closer to those of lizards. Bones involved in feeding and jaw movement were already changing, but they had not reached the extreme mobility found in many living snakes. The body and skull were evolving together, though not necessarily at the same pace. Najash did not capture one instant when a lizard became a snake. No such instant existed. Different parts of the transformation occurred at different times. The body elongated. The forelimbs disappeared. The hind limbs remained longer. The pelvis became smaller. Its connection to the spine was eventually lost. The skull became more mobile. Muscles and nerves changed to control a new style of movement. Genetic systems regulating limb growth were altered. Limb loss was not one event. It was a collection of changes spread across anatomy and deep time. That is why an animal can look unmistakably like a snake while still carrying legs. Evolution does not wait for every feature to change together. Nor does a structure always vanish as soon as its original role becomes less important. Some bones remain as remnants. Others become smaller. A few are given entirely new jobs. The clearest example lies inside the heads of mammals. Two tiny bones that allow humans to hear were once part of the jaw joint of our distant ancestors. For that transition to occur, bones used in biting had to stop carrying the force of a bite. New fossils revealed how that became possible. Number 6. Dinoconodon and Pterodocodon. Place a finger just in front of your ear and open your mouth. The movement you feel comes from the mammalian jaw joint. The lower jaws is formed mainly from one bone called the dentary. It connects with a skull bone called the squamosal. Inside the nearby middle ear are three far smaller bones. The malleus, incus, and stapes transmit vibrations from the eardrum toward the inner ear. This arrangement seems ordinary because every living mammal possesses some version of it. The distant ancestors of mammals were built differently. Their lower jaws contained several bones. The main jaw joint formed between the articular, at the back of the jaw, and the quadrate, in the skull. Those bones correspond to the malleus and incus of mammals. At some point in evolutionary history, the old jaw joint became part of the hearing system. The problem was mechanical. Bones cannot function as delicate sound transmitters while also absorbing the full pressure produced by biting. Before the old joint could become an ear, another joint had to take over the work of the jaw. Fossils of mammal relatives preserved parts of that transition. Some animals possessed two jaw joints at the same time. The old articular quadrate connection remained. A new contact had also formed between the dentary and squamosal. For a period, both joints shared the load. That arrangement gave the older bones a chance to become smaller and less important for supporting the jaw. The details were difficult to observe because the bones were tiny. They were easily crushed or lost during fossilization. In many specimens, they remained hidden inside solid rock. High-resolution scanning changed what could be seen. In Jurassic fossils from China, researchers described two small mammal relatives named Dinoconodon and Pterodocodon. Dinoconodon preserved a double jaw joint. The newer dentary squamosal joint was already present and capable of carrying force. The older articular-quadrate joint remained connected, but its mechanical role appears to have been weakening. It had not yet become a fully separated mammalian middle ear. It was no longer solely responsible for biting. Pterodocodon preserved a different condition. Its postdentary bones were more closely associated with hearing and less involved in supporting the jaw. Taken together, the fossils captured separate stages in the release of the old joint from its original function. The transition involved more than bones moving from one place to another. First, the forces passing through the jaw had to change. As the dentary enlarged and the new joint became stronger, the smaller bones at the rear could be reduced. Smaller bones respond more easily to vibration. Connections that once stabilized the jaw could become thinner. Eventually, the old joint could operate as a suspended chain transmitting sound. Other fossils preserved additional arrangements. Liaoconodon, another mammal relative from China, had middle ear bones separated from the main jaw, but still connected by an ossified form of Meckel's cartilage. The hearing system was no longer functioning as a jaw joint, but it had not become completely independent from the lower jaw. This was not a defective or unfinished animal. It was a workable anatomical condition. Different mammal lineages appear to have explored several arrangements of the jaw and middle ear. Some features may have evolved more than once. Others may have changed in different sequences. The process was not perfectly linear. The broader transformation is still clear. The articular became the malleus. The quadrate became the incus. Bones once used to connect the jaw to the skull became part of a system capable of detecting faint vibrations. That change helped mammals hear higher frequencies. For small nocturnal animals living among dinosaurs, improved hearing may have been especially valuable. It could reveal insects moving through vegetation. It could reveal predators approaching in darkness. It could reveal calls from members of the same species. No entirely new ear bones had to appear. Evolution worked with bones that were already present. It shifted the forces passing through them. It changed their size. It altered their connections. A feeding structure became a sensory structure. That transformation occurred inside animals small enough to fit in a human hand. The next discovery involved the reorganization of an entire body. A group of four-legged mammals began spending more time in water. Millions of years later, their descendants would become whales. The first step was taken by an animal that looked more like a tiny deer. Number 5. Indo-Hyas. A blue whale can reach the length of a large aircraft. Its body is supported by water. The front limbs have become flippers. The hind limbs have disappeared from the outside, leaving small bones buried within the body. The tail produces powerful vertical strokes. The nostrils have moved toward the top of the skull. Nothing about that anatomy immediately suggests a relationship with hoofed mammals walking on land. Genetic evidence places whales within the even-toed ungulates, the group that includes deer, pigs, cattle, and hippopotamuses. The fossil record had already revealed several early whales with legs. Pachycetus lived mostly on land, but had ear structures associated with cetaceans. Ambulocetus possessed strong limbs and could move through both land and water. Later forms became increasingly aquatic. Their hind limbs reduced. Their bodies lengthened. Their tails became more important for propulsion. A question remained. What happened before the earliest recognizable whales? Researchers examining Eocene fossils from India studied a small animal called Indohyus. It belonged to an extinct group known as the rowellids. The body was lightly built. The legs were slender. The feet ended in small hooves. At first glance, Indohyus resembled a miniature deer or chevrotain. Several details did not fit a completely terrestrial lifestyle. One was located in the ear region. Whales possess a distinctive thickened structure called the involucrum, formed from part of the bone surrounding the middle ear. It is one of the anatomical features used to identify early cetaceans. Indohyus had a similar thickened region. It was not identical to the ear of a modern whale, but it supported a close evolutionary relationship. The limb bones offered another clue. They were unusually dense. Dense bones act as ballast. Animals that walk along river bottoms or remain submerged in shallow water often carry heavy skeletons that reduce buoyancy. Hippopotamuses use dense bones in this way. So do several other semi-aquatic mammals. The compact bones of Indohyus suggested that it regularly entered water. Researchers also examined the chemistry of its teeth. The ratio of oxygen isotopes preserved in enamel reflects the water an animal consumed and the environment in which it lived. The values from Indohyus were consistent with a life strongly connected to aquatic habitats. Its teeth did not show the same feeding pattern as those of early predatory whales. That distinction changed the likely order of events. The close relatives of early whales may have entered the water before they began hunting aquatic prey. Wading, hiding underwater, or feeding along river margins could have come first. A major shift in diet may have followed later. A loose, modern comparison can be found in the African water chevrotain. When threatened, this small hoofed mammal may enter a river and remain submerged. Indohyus was not the same animal. Its behavior cannot be reconstructed simply from appearance. The comparison still shows how a hoofed mammal could begin using water without immediately turning into a specialized swimmer. Indohyus was not the first whale. It has not been proven to be the direct ancestor of every cetacean. Phylogenetic analyses place rowellids close to the cetacean branch, probably as a sister group. That position is exactly what makes the fossil useful. It preserves traits near the beginning of the transition, without needing to stand directly on the ancestral line. The animal also demonstrates that ecological change can come before dramatic anatomical transformation. Indohyus still had legs and hooves. It could still move on land. Its ears, heavy bones, and tooth chemistry reveal an increasing dependence on water. Later cetaceans carried the transition much further. Their limbs became adapted for swimming. The pelvis detached from the spine. The hind legs shrank. The nostrils moved backward. The tail became the main source of thrust. A land mammal was gradually reorganized into an animal that would spend its entire life at sea. This return to water was possible only because vertebrate ancestors had made the opposite transition hundreds of millions of years earlier. Before a mammal could walk back into the ocean, fish had first developed the anatomical foundations of feet. Acanthostega revealed that those feet did not begin as tools for walking on land. Number 4. Acanthostega. The familiar story of vertebrates leaving water is simple. A fish reaches the edge of a drying pool. Its fins become stronger. It drags itself onto land. Over generations, the fins turn into legs. The animal begins walking. That story places feet after the move onto land. Acanthostega reversed the order. Its fossils were discovered in late Devonian rocks in eastern Greenland. The animal lived about 365 million years ago. It belonged near the early history of tetrapods, the vertebrate group that includes amphibians, reptiles, birds, and mammals. Acanthostega had limbs ending in digits. That already separated it from an ordinary fish. Each front limb carried 8 digits. The familiar five-fingered arrangement of later tetrapods had not yet been established. Other early tetrapods added to the pattern. Ichthyostega had 7 digits on the hind limb. Tularpeton had 6. Early limb evolution experimented with several numbers before the 5-digit pattern became dominant. The more important question was where those digits were being used. Acanthostega retained internal gills. Parts of the branchial skeleton that supported them were preserved. Its tail also remained well suited to swimming. The limbs were not built like the weight-bearing legs of a land animal. The joints allowed movement, but the arrangement of bones and muscles was poorly suited to holding the body clear of the ground. The rib cage was relatively weak. On land, strong ribs help support the organs and prevent the torso from collapsing under gravity. In water, buoyancy performs much of that work. Acanthostega remains strongly dependent on an aquatic environment. Why then did it have digits? The limbs may have helped it move through shallow water filled with plants. A broad hand with several digits could press against vegetation, branches, or the bottom. The animal may have raised its head above the surface, while the rest of the body remained supported by water. It may have maneuvered through narrow channels. It may have stabilized itself in currents. The exact behavior cannot be observed. No single reconstruction is certain. The anatomical conclusion is stronger. Digits existed before efficient terrestrial walking. That changes the meaning of the first feet. They were not necessarily created after vertebrates reached land. They developed in fish-like animals that were still living in water. Only later were they modified to carry the body against gravity. Acanthostega also exposed another difficulty in interpreting fossils. Many known specimens may not have been fully mature. Studies of bone growth suggest that some individuals were juveniles whose skeletons had not finished developing. An adult may have been somewhat stronger. Even so, the gills and overall anatomy still point to a heavily aquatic lifestyle. The transition was a mosaic. Digits appeared. Gills remained. The tail continued to provide propulsion. The limbs acquired joints. The torso was not yet an efficient weight-bearing bridge. Different parts of the body crossed the boundary at different times. Acanthostega showed that a foot could exist before the first effective step on land. The next question was where the structure of that foot had begun. Scientists needed a fossil slightly older and more fish-like. They searched rocks of the correct age and environment between known lobe-finned fish and the earliest tetrapods. In the Canadian Arctic, they found an animal with scales, fins, and gills. Inside one fin was a jointed skeleton capable of bending like a primitive wrist. Number 3. Tiktaalik. Tiktaalik was not found by chance. Its location was predicted. Scientists knew that fish with some tetrapod-like traits lived around 380 million years ago. Recognizable early tetrapods appeared several million years later. A fossil preserving an intermediate combination should come from rocks between those ages. The environment mattered too. The transition was expected to have occurred in rivers, floodplains, or shallow freshwater channels. Geological maps identified exposed late Devonian rocks on Ellesmere Island in the Canadian Arctic. Today, the region is cold and treeless. During the Devonian period, it lay in a warmer climate. River systems wound across a broad floodplain. Field teams traveled there and searched the layers, season after season. Eventually, part of a flattened skull appeared in the stone. The animal was named Tiktaalik rosier. It was approximately 375 million years old. Tiktaalik was clearly a fish. Its body carried scales. It had fins with fin rays. It possessed gills. Its jaws and much of its skull resembled those of other lobe-finned fish. The top of the skull, however, was broad and flat. The eyes faced upward. The head was separated from the shoulder girdle, creating a movable neck. Most fish cannot turn the head independently because the skull and shoulders are joined. Tiktaalik could. Its ribs were enlarged. They may have helped stabilize the body and support breathing movements. The most important evidence was hidden inside the pectoral fins. A fin may look simple from the outside. Beneath the surface, lobe-finned fish possess an internal skeleton. In Tiktaalik, that skeleton was organized in a pattern approaching the limb of a tetrapod. Closest to the body was one large bone comparable to the humerus. Beyond it were two bones corresponding to the radius and ulna. Farther outward were smaller bones connected by joints. They were not true fingers. Fin rays still surrounded the edge. The internal joints could nevertheless bend. The distal part of the fin could press against a surface while the elbow flexed. It functioned like an early wrist. Mechanical studies suggest that Tiktaalik could support part of its front body with the fins. That does not mean it walked across dry land like a salamander. The fin rays remained important. The animal was still adapted to water. A more cautious reconstruction places it in shallow channels. It could brace against the bottom. It may have raised its head above the surface. It might have pushed through mud, vegetation, or water too shallow for ordinary swimming. In poorly oxygenated conditions, lifting the head to breathe air could have been useful. The flat skull and upward-facing eyes may have allowed it to watch the surface or the bank, while staying mostly submerged. Each major feature had roots in fish anatomy. The neck developed partly by separating the shoulder girdle from the skull. The wrist-like joint came from bones already present in a lobe-finned fin. Stronger ribs expanded structures fish already possessed. The transition did not require a completely new body to appear. It changed the relationships among existing parts. Tiktaalik became famous because it matched a scientific prediction. Researchers used evolution and geology to identify where a certain combination of features should be found. The fossil then appeared in rocks of the expected age and environment. That did not make Tiktaalik the one ancestor that crawled onto land and produced every later tetrapod. It was one branch close to a broader transition. Other related animals may have developed similar abilities. Many left no known fossils. The evolutionary tree was branching. It was not marching forward in a line. Tiktaalik and Acanthostega also preserved different stages. Tiktaalik had a wrist-like fin without digits. Acanthostega had digits, but remained strongly aquatic. Together, they show that the limb was assembled before efficient walking on land. The first wrist developed underwater. The first digits developed underwater. Only later were those structures adapted to carry a body across land. The same principle would reappear in the history of flight. Wings did not begin as complete flying structures. Feathers did not arrive at the instant an animal became a bird. For more than a century, one fossil stood at the center of that argument. It had the wings and feathers of a bird. It also had teeth, claws, and the long bony tail of a dinosaur. Number 2. Archaeopteryx. In the 19th century, limestone was quarried near Solnhofen in southern Germany. The rock was fine-grained and could be split into smooth, thin slabs. It became valuable for printing. The same stone preserved animals from a late Jurassic lagoon about 150 million years ago. The water was calm. The bottom contained little oxygen. Bodies that sank into the lagoon were less likely to be disturbed by scavengers. Fine sediment settled around them, recording details that would normally vanish. Jellyfish, insects, fish, and flying reptiles survived in remarkable detail. In one slab, a single feather appeared. Soon afterward, a skeleton was found with impressions of feathers around its arms and tail. The animal was named Archaeopteryx. Its importance was immediately obvious. Charles Darwin had published On the Origin of Species only a few years earlier. Critics argued that the fossil record lacked organisms linking major groups. Archaeopteryx appeared to combine two of them. The wings carried long flight feathers, arranged much like those of modern birds. The feathers were asymmetrical, with the shaft closer to one edge. That shape helps produce an aerodynamic surface. The tail also supported paired feathers. Beneath the plumage was a skeleton unlike that of any modern bird. The jaws contained teeth. The tail consisted of a long series of separate vertebrae. 3 clawed fingers extended from each hand. The body retained several traits associated with small theropod dinosaurs. Archaeopteryx was not simply a bird with a few reptilian details. It was a mosaic. Some regions of the body were strongly avian. Others remained closer to non-avian dinosaurs. The discovery changed the debate. Birds and reptiles could no longer be treated as groups divided by an unbridgeable anatomical gap. Archaeopteryx created new questions at the same time. Could it truly fly? The feathers were suitable for producing lift, but the skeleton lacked several features used by strong, modern flyers. The breastbone was not enlarged into the deep keel that anchors powerful flight muscles in many living birds. The shoulder joint and wing movement may also have differed. Some researchers argued that Archaeopteryx mainly glided from trees. Others proposed that it could take off from the ground. Studies of feather orientation, joint movement, and bone strength have produced several models. The safest conclusion is that Archaeopteryx was capable of some form of aerial movement. It was not necessarily as efficient as a modern bird. Its exact flight style remains debated. The skull offered another source of evidence. CT scans allowed researchers to reconstruct the brain case and inner ear. Regions associated with vision, balance, and coordination were enlarged compared with many reptiles. The inner ear had proportions useful for controlling movement in 3 dimensions. The nervous system already possessed several features valuable in the air. The body covering was also more extensive than the classic image of 2 feathered wings. Exceptionally preserved specimens showed long feathers on the legs and other regions. Not every feather was shaped specifically for powered flight. Some may have contributed to display, insulation, or control during movement. This suggested that feathers were evolving in several functional contexts at once. Even the position of Archaeopteryx on the evolutionary tree became uncertain. For many years, it was called the first bird. But first bird is not a simple scientific category. New feathered dinosaurs shared features once thought unique to Archaeopteryx. Depending on the data and method used, the animal sometimes appeared at the base of the bird lineage. In other analyses, it moved closer to Deinonychosaurs. Later studies often returned it to Aviale, the branch containing birds and their closest extinct relatives. Those changes did not weaken the fossil's importance. A transitional fossil does not need to be the direct ancestor of a living group. It does not need to occupy one permanent position in every analysis. Its value lies in the combination of traits it preserves. Archaeopteryx proved that flight feathers, dinosaur-like teeth, clawed hands, and a long bony tail could exist in the same body. It recorded a stage when the avian form had not yet been fully assembled. For decades, Archaeopteryx seemed almost alone. There were dinosaurs without known feathers and birds with feathers, with few fossils between them. Perhaps Archaeopteryx was an isolated experiment. Perhaps feathers were limited to a narrow branch already close to birds. Then, paleontologists began examining fossils from Liaoning Province in northeastern China. The rocks preserved not one feathered dinosaur, but many. Some could not fly. Some carried simple filaments rather than modern flight feathers. Some had long feathers on all 4 limbs. One was a large relative of tyrannosaurs. Archaeopteryx was no longer standing alone. It belonged to an entire feathered world. Number 1. The feathered dinosaurs of Liaoning. We return to the stone slab from the opening. A small dinosaur lies on its side. The bones are dark against pale rock. Around the neck, back, and tail is a faint covering of filament-like structures. When fossils like this first appeared from Liaoning, their meaning was uncertain. Soft tissues can decay into misleading forms. collagen fibers may separate from the body. Plants and microorganisms can be buried beside an animal. A dark halo around a skeleton is not automatically a coat of feathers. Liaoning did not produce only one specimen. The region contained an entire fossil ecosystem known as the Jehol biota. During the early Cretaceous period, lakes spread across the landscape. Volcanoes erupted nearby. Fine ash fell into the water and mixed with sediment. Animals buried rapidly on the lake floor could be sealed before the bodies were destroyed. The deposits preserved fish, amphibians, mammals, insects, plants, birds, and dinosaurs. Skeletons remained articulated. Stomach contents survived. In some cases, skin, membranes, and body coverings left visible impressions. One of the first dinosaurs to transform the discussion was Sinosauropteryx. It was a small theropod with a long tail and short forelimbs. It was not close enough to modern birds for its covering to be dismissed as an ordinary bird feature. Along the head, neck, back, and tail were narrow, filament-like structures. They did not resemble the broad flight feathers of a bird. They formed a simple, fuzzy coat. If those structures were related to feathers, then feathers had existed in a dinosaur that could not fly. That challenged the idea that feathers evolved only for flight. Critics proposed that the filaments were damaged collagen fibers from beneath the skin. Appearance alone could not settle the argument. More fossils followed. Sinornithosaurus, a small dromaeosaur relative, preserved structures with branching patterns. Instead of single, unbranched strands, some filaments divided. They resembled intermediate stages predicted by models of feather development. In living birds, feathers begin as tubular growths in the skin. Developmental changes produce branching barbs, smaller barbules, and eventually a central shaft. The Liaoning fossils appeared to preserve several levels of structural complexity across different dinosaurs. Other specimens carried fully developed pinnaceous feathers. These had a central shaft and organized veins, broadly comparable with the feathers of birds. One dromaeosaur fossil preserved clear feather attachment along the arm. Another showed long feathers on the tail. The simple division between feathered birds and scaly dinosaurs was collapsing. Perhaps the most visually unusual animal was Microraptor. It was a small dromaeosaur with long pinnaceous feathers on the arms. Similar feathers extended from the hind limbs. The tail supported another aerodynamic surface. Microraptor is often described as having 4 wings. The forelimbs and hind limbs did not function as 4 identical bird wings, but all 4 carried long feathers capable of influencing airflow. Researchers tested several possible arrangements. Some placed the hind limbs below the body. Others extended them outward. Wind tunnel experiments and computer models produced different results, depending on the assumed posture. The animal was probably capable of gliding. It may also have used flapping movements. Its exact technique is still debated. The larger meaning is more secure. Early aerial movement involved more anatomical experimentation than the modern 2-winged bird body suggests. Long feathers appeared on the hind limbs before becoming reduced in later birds. Different branches explored different ways of producing lift, stability, and control. Feathers were not waiting for one final design. They were being used in several configurations. Liaoning also showed that feather-like coverings were not limited to very small dinosaurs. Eutyrannus was a large tyrannosauroid. The known specimens included animals several meters long. They were far heavier than the small feathered theropods discovered earlier. Around parts of their skeletons were long, filamentous structures. Eutyrannus lived in an environment with cool, seasonal conditions. A coat of filaments may have helped retain heat. This does not mean every large dinosaur carried a thick covering. Skin impressions from other species show scales across substantial parts of the body. Feather distribution differed among lineages. It may also have changed with body size, age, and environment. Eutyrannus still expanded the known range. A feather-like covering was compatible with a large, predatory dinosaur. The fossils eventually began revealing color. Under powerful microscopes, researchers identified microscopic structures inside fossil feathers and filaments. They resembled melanosomes, the pigment-containing bodies found in modern feathers. Different shapes and arrangements are associated with different colors. By comparing the fossils with living birds, scientists reconstructed parts of the appearance of several dinosaurs. Sinosauropteryx appears to have had alternating light and dark bands along the tail. Other species carried black, gray, reddish, or iridescent patterns. Color was not simply decoration added to an illustration. It strengthened the identification of the structures as feathers. A collagen fiber would not be expected to contain pigment bodies, arranged like those in a biological covering. The patterns also hinted at function. A banded tail could serve in display or recognition. Dark and light regions might provide camouflage. Bright feathers could signal age, health, or reproductive condition. Long before feathers became essential flight surfaces, they may have carried information. Insulation was another likely early role. A simple filamentous coat traps air near the skin. Small animals lose heat quickly because their surface area is large compared with body volume. A covering of protofeathers could help control temperature. Feathers may also have protected the skin. They may have covered nests. They may have helped adults regulate the temperature of eggs. Broad feathers could improve balance while running or climbing. They might produce drag when an animal jumped, turned, or descended from an elevated position. Natural selection could then improve those aerodynamic effects. This is exaptation. A structure that evolved in one context is later recruited for another. Feathers did not need to begin as perfect flight equipment. They could begin as insulation. They could become more elaborate for display. Broad veins could produce small aerodynamic benefits. Those benefits could become useful during jumping, braking, gliding, or running. In some branches, feathers and forelimbs were eventually integrated into powered wings. Most feathered dinosaurs did not follow that path. Many remained unable to fly. Others developed aerial abilities that left no living descendants. One branch survived the end of the Cretaceous and diversified into modern birds. Birds are therefore not merely descended from dinosaurs, as though they left the group behind. Under evolutionary classification, birds remain dinosaurs. A sparrow is a living dinosaur. So is an eagle. So is an ostrich. So is a penguin. The asteroid impact, 66 million years ago, eliminated every known non-avian dinosaur. One feathered dinosaur branch survived. The Liaoning discoveries changed more than the way prehistoric animals were drawn. Before them, popular reconstructions often showed almost every dinosaur with similar reptilian scales. Afterward, paleontologists had to ask which dinosaurs carried feathers. They had to ask what type of feathers they possessed. They had to ask where the feathers grew. They had to ask what functions those feathers served. The question was no longer whether birds were closely related to dinosaurs. It became a question of sequence. Which avian features appeared first? Which developed for reasons unrelated to flight? Which branches experimented with combinations that later disappeared? The faint halo around the skeleton from the opening did not mark the instant a dinosaur became a bird. It preserved one stage in a much longer history. Simple filaments appeared before modern feathers. Feathers became complex before powered flight. Long feathers spread across limbs that were not yet arranged like those of living birds. Flight-related anatomy emerged across several branches. The boundary between dinosaur and bird was not crossed in a single leap. Feature by feature, the boundary disappeared. These 10 discoveries span more than 500 million years. At first, they seem to tell unrelated stories. One concerns an animal pressed into an Ediacaran seafloor. Another concerns the shell of a turtle. Others involve a snake's legs, a mammal's ear, a whale's return to water, the first digits, and the origin of feathers. The same pattern connects them. Evolution works with structures already present. Kimberella showed that complex animal organization began earlier than the abundance of Cambrian skeletons once suggested. Dickinsonia showed that chemistry can identify an animal, even when its anatomy has no close modern comparison. Odontoceles revealed a shell assembled through changes to ribs, belly bones, vertebrae, and muscles. Najash preserved a snake body before the last external limbs had disappeared. In early mammal relatives, the old jaw joint was not discarded. Its bones became part of the ear. Indoheus showed that a lineage could enter the water before it looked or fed like a whale. Acanthostiga possessed digits before it could walk efficiently on land. Tiktaalik carried the architecture of a wrist inside a fin. Archaeopteryx combined flight feathers with teeth, claws, and a long dinosaur tail. The fossils of Liaoning showed that feathers existed in many forms before modern birds and powered flight were complete. These organisms were not defective versions of animals waiting to become modern. Each was adapted to its own environment. Each combination of traits functioned in a living body. The fossils matter because they preserve combinations that later vanished. They reveal that transformations, which appear sudden from a distance, were built from smaller changes spread across anatomy and time. A rib can broaden. A leg can shrink. A jawbone can lose its mechanical load. A fin joint can become more flexible. A filament can branch. A structure can serve one purpose before being recruited for another. No single fossil contains the complete history of a major group. When many discoveries are compared, however, the gaps begin to narrow. The result is not a ladder leading toward the present. It is a branching tree filled with experiments, extinctions, and unexpected survivors. Most prehistoric animals left no fossils. Their bodies were eaten, scattered, or destroyed before burial. Even when fossilization occurred, erosion could erase the evidence before anyone found it. What remains is a fraction of a fraction. A tooth, a feather impression, a joint hidden inside stone, a chemical trace in a film of organic material. Sometimes the importance of a fossil is not recognized when it is first discovered. It may wait in a museum drawer until a new scanner reveals its bones. It may require another specimen from another continent. It may depend on a method of chemical analysis that has not yet been invented. The fossil record is incomplete. That does not mean the investigation has failed. It means the history of life is still being tested. Somewhere inside an exposed cliff, a quarry wall, or an unopened stone slab, another detail may already be waiting. A detail that does not fit the story we currently believe. And when someone finally recognizes it, evolution may need to be rewritten again.